Yes, sir.
Okay, good afternoon, students. So, today we are going to discuss about glycogenesis. This is the first topic in glycogen metabolism. So, we know that every organism to live needs energy. Then, what is the source of energy for all living organisms? It is the light energy of sun. And during photosynthesis, it is converted into chemical energy. And this chemical energy is nothing but glucose. Thus, for all the living organisms, though it is synthesized, glucose is synthesized by the plants. Plants are eaten by herbivores, herbivores are eaten by carnivores, and both herbivores and carnivores, as well as plants, are eaten by omnivores. That means the ultimate producers in the food chain are plants, where glucose is present. Thus, directly or indirectly, all living organisms on the surface of the earth, mainly the higher organisms, depend for their food material on glucose. So it is the immediate source of energy. Whenever you feel weakened, when you are taken to hospital to the doctor, what does the doctor do? The doctor gives what we call glucose through a bottle to directly to your blood, isn't it? So thus we get energy from glucose only. All cells need constant supply of glucose. That means every cell is performing a number of metabolic activities. We have already seen glycolysis, Krebs cycle, electron transport. For all these reactions, we require energy, isn't it? So here, for the energy production, what happens? The cells require glucose continuously. And coming to the two important cells, one is RBC, which carries oxygen and carbon dioxide during respiration. The other is the cells of the brain. For these two cells, glucose is the only source. They do not depend on other sources of energy. They cannot utilize fats. Fats are also another source of energy, but they cannot utilize. That means if a brain needs energy, you have to supply only glucose, isn't it? And coming to the other things, cells of human beings and other vertebrates derive glucose. How they derive glucose? They derive glucose from blood. So, for all the cells of the body, the glucose is supplied only through the blood. Then, how the blood gets the glucose? It gets only from food, mainly from food. And secondly, from a process called as glycogenolysis, that is breakdown of glycogen. And thirdly, from a process called as gluconeogenesis. So whatever the cells require, this is the cell requires glucose, isn't it? So this glucose is supplied by blood. And how the blood receives? From diet, that means through food. And second source is by the breakdown of glycogen, and the third source is gluconeogenesis. So that glucose is released into the blood. From there, it is circulated to all cells of the body. Then, what are these glycogen and gluconeogenesis are? And we have already seen just now seen that source of glucose are diet glycogen and gluconeogenesis. And what about this glycogen? What actually happens is, though there are three sources, diet, glycogenolysis, and gluconeogenesis, what scientists have discovered that during fasting or starvation, cells may not get glucose from diet or gluconeogenesis. When you are under fasting, you do not eat food, so that the blood did not receive glucose through food. Then what about the other thing? Gluconeogenesis. Gluconeogenesis is nothing but conversion of certain non-carbohydrate sources like amino acids to glucose. So glucose can be formed from certain amino acids called as glucogenic amino acids. Glucogenic amino acids. So this type of production of glucose from non-carbohydrate sources is called as gluconeogenesis. But when we are fasting or undergoing starvation, even the animals also, they do not get glucose from food or by gluconeogenesis. Then how come they survive? Dietary source of glucose and its precursors such as starch and disaccharides depend on diet, which body may not get during starvation. Whereas gluconeogenesis responds slowly to falling glucose levels. So these two are eliminated. Then what about this? Therefore, body has developed a mechanism for storing a supply of glucose in a rapidly mobilizable form called as glycogen. So, out of these three resources, glycogen is the immediate source of glucose. Glycogen is the immediate source of glucose because it can be rapidly mobilizable, which can be broken down and be supplied through blood to the glucose. That is the reason why all animals, all animals store glucose in the form of glycogen in the liver and muscle. Whereas after photosynthesis, when glucose is formed, this is stored as starch in plants, that is in animals, the same glucose is stored as glycogen. Okay. Whenever blood glucose falls down, whenever blood glucose falls down, just a minute, you can have now. We actually has our fingers in it. So whenever blood glucose falls down, glycogen is immediately broken down to release glucose. So liver stores glucose as glycogen, and now the cells require glucose because we are undergoing fasting. We have eaten food, we have taken food in the morning 7 o'clock, and you did not bring lunch to the college, and the classes are till 5 o'clock, and Samuel sir has taken extra class till 7 o'clock. That means 7 to 7 you do not have any food source at all. So though you are not fasting, though you are not starving, you are said to be in, unintentionally, or you are going to starve. In that case, what happens? The cells require glucose, which is not supplied. All the glucose that is present in the blood is supplied, and it is utilized by the cells from the morning to evening. Then what about the remaining time? By the time you reach home, you cannot get any glucose unless otherwise you go to a shop and eat something. So, but the body, what happens is the food that we take, glucose, if it contains glucose, the body utilizes some of it, and some of it is stored as glycogen. So, from the stored glycogen reserves, it is broken down immediately and it is released to blood, and from the blood it reaches the cells, and thereby glucose is utilized. Thus, synthesis and breakdown of glycogen are two important mechanisms of glycogen metabolism. Here glucose is converted into glycogen. This is called as synthesis, formation of or synthesis of glycogen. And the other thing is glycogen, whenever glucose is required, it is broken down to release glucose. This is called as breakdown. So thus there are two processes. One is synthesis of glycogen, the other is breakdown of glycogen. Synthesis of glycogen is called as glycogenesis, glycogenesis, and breakdown of glycogen is called as glycogenolysis. So these two are the important aspects of glycogen metabolism. So before going to that, let me explain the structure of glycogen. So what actually glycogen is? Glycogen is a highly branched polysaccharide. You know that what is a polysaccharide? Polysaccharide is a carbohydrate which is having not more than 10 glucose or fructose or any other monosaccharide residue. And here it is a polysaccharide made up of alpha dextroglucose, alpha D glucose. Okay, so polysaccharide means if this is alpha D glucose, another glucose is added, another glucose is added, and what happens? A long chain of glycogen is formed. Okay, and between these residues, the bond is between the first carbon of one glucose, the fourth carbon of another glucose. So it is called as alpha 1,4 glycosidic bond. So it has alpha 1,4 glycosidic bonds. But actually what happens is glycogen is not a linear structure. It is a branched polymer. That means branches arise from at certain points. And here also another branch arises. And here also another arises. So these are the branching points. These are the branching points from which another polymer of glucose starts. So here in the branching points, what the scientists say is the bond is between the first carbon of one glucose and the sixth carbon of another glucose. Thus it is called as an alpha 1,6 glycosidic bond. So at branching points, for every 10 residues, a chain of glucose residue branches off by the presence of 1,6 glycosidic bonds, and a number of branches are formed. And even from this branch, another branch can be formed. Another branch can be formed. Thus, the glycogen is a highly branched structure. Okay, so this type of structure, the elementary particle of glycogen, which is formed because of these branchings, is called as a beta particle. And its size is 21 nanometers in diameter, and it consists of 55,000 glucose residues. So what is the number of glucose residues present in glycogen? It is 55,000. Okay, and the molecular weight is said to be 10 to the power of 8 daltons. And glycogen makes about 1 to 2% of muscle tissue and 10% of liver tissue by weight. I have already told you, glycogen is stored only in muscle and liver, not elsewhere. And in the muscle, the amount of glycogen is 1 to 2%. The amount of glycogen in liver is 10%. Okay. Coming to the glycogen metabolism, the first event is glycogenesis, that is synthesis of glycogen. How it takes place, let us see. Biosynthesis of glycogen, that is formation of glycogen simply, is called as glycogenesis. How it is synthesized? We have seen that glycogen is a polysaccharide made of alpha D glucose. Thus, it is synthesized from molecules of alpha D glucose. So, where actually the process of glycogenesis takes place? Does it take place in any cell organelle or nucleus? No, it takes place in the cytoplasm itself. So, glycogen metabolism, mainly glycogenesis, takes place in the cytoplasm, and it requires the synthesis of glycogen requires energy. Okay, so it requires energy which is supplied by ATP and also by UTP. We know adenine, guanine, cytosine, thymine, and uracil. These are the nitrogen bases. And adenine gives rise to adenosine triphosphate, guanosine triphosphate, cytosine triphosphate, thymine triphosphate, and uridine triphosphate. And these two are the sources of energy during the synthesis of glycogen. So glycogenesis, this very process of synthesis of glycogen draws energy by the breakdown of ATP and by the breakdown of UTP. And coming to the steps, various steps involved in the synthesis of glycogen, the first step is synthesis of uridine diphosphate glucose. We simply call it as UDPG. In shortcut, uridine diphosphate glucose. Okay, what actually happens during the synthesis? Here, this is blood. Okay, so blood supplies all things to the cells. The cell requires vitamin B1. How it is supplied? From the blood. How the blood gets? From the food. So from the food, vitamin B1 goes to blood. From there, it goes to the cells. Likewise, blood fatty acids the cell requires. It is supplied by blood. From where? Through the food. Amino acids the cells require. It is supplied in this way. Likewise, glucose is also supplied through diet and blood to the cells. So let us see, the cells are said to be drawing glucose continuously from the blood. What is the glucose level of blood? Glucose level of blood. Scientists say for every 100 ml of blood, there is 90 mg of glucose. What happens? 10 mg of glucose is utilized in the first hour. In the second period, you have utilized another 10 mg of glucose. By the time the college is over, it is gradually dropping down to 30 mg. And what happens? The amount of energy that is required to supply all the cells of the body, that is glucose, is gradually depleting. Now what happens here? You already have stored glycogen, glucose in the form of glycogen in the liver. Now, a hormone, whenever the blood glucose level drops down, pancreas releases a hormone called as insulin. So this insulin is responsible for breakdown of, sorry, this, this insulin is responsible, sorry, whenever the body is requiring what we call glucose, glycogen is broken down to release glucose. So this glucose is supplied to the cells, and this process is called as glycogenolysis. For this, a hormone called as glucagon is required. So glucagon is responsible for the breakdown of glycogen. Then we are discussing about glycogenesis, that means synthesis of glycogen. So glucose we have taken into blood. So lots of glucose is taken. Somebody has brought me some, what we call this, because this is summer season. I have got so many mangoes. Okay, and I have so many sweets also. I have, so every day I'm eating all this, and the amount of glucose in the blood is gradually increasing. So which is actually 90 mg per 100 ml, it is now increased to 190 mg. Then what happens? In the blood, all the other constituents also should be there. So that here it has to be reduced to 90 again. Then what about the remaining 100? So this 100 mg of glucose, which is excess in blood, is taken up by the liver, where the liver converts glucose to glycogen. So who does this conversion of glucose to glycogen? This is accomplished by a hormone called as insulin. Thus, there are two hormones for glycogen metabolism. One is for the breakdown of glycogen, which is called as glucagon. The other is for the synthesis of glycogen, which is called as insulin. Okay, so under the influence of the hypoglycemic hormone insulin, what is hypoglycemic? Decrease in blood glucose level. It is already I have told you, it is 190 mg. It has to be decreased to normal level, 190 mg. So decrease in blood glucose level is called as hypoglycemia, and this insulin is responsible for the decreasing blood glucose level by converting glucose to glycogen. Okay, hence it is called as hypoglycemic hormone. So under the influence of this hormone, it is taken up by the liver cell. Glucose is taken up by the liver cells from blood. Immediately what happens? This is blood, glucose is present, and this glucose goes to cells. What happens here? There are 100 molecules here previously. Now here there are only 10, 90 molecules are here. So there is a concentration gradient being established. There is every chance for the glucose molecules to come back into the blood again. In order to prevent this, what happens is glucose is converted into glucose 6-phosphate. Glucose is converted into glucose 6. How come it is possible? So glucose is a 6-carbon compound. It is a 6-carbon compound, and to the 6th carbon, phosphate is added. Phosphate is added. So that what happens now? Glucose is converted into glucose 6-phosphate. Now what happens here? This is mainly because of the fact that the cell membrane is permeable to glucose, but cell membrane is impermeable to glucose 6-phosphate. So hence, whenever the glucose is converted into glucose 6-phosphate, it cannot be sent out. We can say simply that it is trapped. So glucose is immediately phosphorylated to glucose 6-phosphate, so that it is trapped in the liver cell. Then glucose 1,6-phosphate is generated from glucose 6-phosphate. So glucose is converted into glucose 6-phosphate, and from this glucose 6-phosphate, glucose 1 phosphate is formed. But before glucose 1 phosphate is formed, what happens is there is an obligatory intermediate reaction, where glucose 1,6 bisphosphate is formed first, followed by the formation of glucose 1 phosphate. So this is the one which is required for the synthesis of glycogen. Okay, so what actually takes place here? Glucose is converted into glucose 6-phosphate. Glucose 6-phosphate is converted into glucose 1,6 bisphosphate, and which is again converted into glucose 1 phosphate. And this is accomplished by an enzyme called as phosphoglucomutase. So for all these steps, phosphoglucomutase is required. So in the initial case, phosphoglucomutase supplies phosphate to glucose, so that glucose is converted to glucose 6-phosphate, where this phosphate is added to the 6th carbon. Hence it is called as glucose 6-phosphate. Now it is trapped inside the liver cell. Now the same phosphoglucomutase converts glucose 6-phosphate to glucose 1,6 bisphosphate. Glucose 1,6 bisphosphate. And after this, what happens? The phosphate on the 6th carbon is removed, and the phosphate on 1st carbon retained, so that by the under the catalytic activity of phosphoglucomutase, what happens? Glucose 1,6 bisphosphate is converted into glucose 1-phosphate. Thus, ultimately, what happens? Glucose is converted into glucose 1-phosphate. Now, can this glucose 1-phosphate be directly utilized for the synthesis of glycogen? Let us see. Direct conversion of glucose 1-phosphate to glycogen and inorganic phosphate is thermodynamically unfavorable. That means it requires lots of energy, and hence what actually does is glycogen biosynthesis requires an additional exergonic step. It was discovered by a scientist called as Louis Leloir, that it occurs by combining glucose 1-phosphate with uridine triphosphate, so that it is converted into uridine diphosphate glucose. So glucose 1-phosphate combines with uridine triphosphate to form uridine diphosphate glucose. And this uridine diphosphate glucose is the activated form of glucose. It is the activated form of glucose, because it has UTP. It is derived from UTP, means UTP like ATP, UTP also energy rich form. So UDPG's high energy status permits it to donate glycosyl or glucosyl units to the growing glycogen chains. So that is the reason why glucose 1-phosphate is not directly utilized in the synthesis of glycogen. Rather, it is converted to form uridine diphosphate glucose. So who catalyzes this? It is catalyzed by what catalyzes it? It is catalyzed by an enzyme called as UDP glucose pyrophosphorylase. So UDPG in short, is synthesized from glucose 1-phosphate and UTP by an enzyme called as UDP glucose pyrophosphorylase. Okay, next. The second step in the synthesis of glycogen is synthesis of primer. So branching of glycogen molecule is catalyzed by the enzyme glycogen synthase. You know that glycogen is not a linear molecule. It has a number of branches, isn't it? So this branching of glycogen molecule is mediated by an enzyme called as glycogen synthase. However, this enzyme, glycogen synthase, cannot initiate chain synthesis using free glucose as an acceptor of a molecule of glucose from UDP glucose. So it cannot initiate synthesis of these branches or glycogen synthesis immediately by utilizing UDPG. Rather, it has some other thing, it requires some other one, that is what we call it as primer. So it can only elongate already existing chains of glucose and therefore requires a primer. Even in during DNA replication and some other things, what happens? There is a primer to which the nucleotides are added. Here also, a small sequence of glucose residues is required, to which this enzyme adds the other glucose residues, so that elongation of glycogen chain takes place. So this small chain is called as primer. So this has to be synthesized first before glycogen synthesis takes place. Okay. So a fragment of glycogen can serve as a primer in cells whose glycogen stores are not totally depleted. So there are every human being does not deplete glycogen sources completely. One day the other day we will be eating something, and so that liver is having glycogen sources. So this glycogen source is utilized to form what we call a small fragment of glycogen, which acts as a primer. In the absence of a glycogen fragment, what happens in those cells where the glycogen fragment is absent? Then what happens? A protein called as glycogenin can serve as an acceptor of glucose residues from UDP glucose. Okay, so there are two types of primers. One primer is glycogen fragment, a fragment of glycogen itself, where the cells are said to be not depleted of glycogen. Whereas there are the cells where there is no supply of glycogen. Then what happens? This fragment of glycogen cannot be used. Then the body uses what we call a protein. This protein is called as glycogenin. So either glycogenin or a fragment of glycogen act as primers, to which what happens? The UDP glucose supplies glucose residues continuously, so that elongation of chain takes place. Okay. The important step in glycogen synthesis involves the attachment of glucose residue to the tyrosine 194 hydroxyl group of a glycogenin. So this is glycogen, this is the primer. This is glycogen. And here on the 194 place, there is tyrosine residue. Protein means it is made up of amino acids. So there are so many amino acids. The 194th amino acid is tyrosine. To the hydroxyl group of this tyrosine, what happens? The attachment of a glucose residue takes place. Thus, glucose chain elongates and glycogen is said to be formed. And this reaction is mediated by an enzyme or catalyzed by an enzyme called as tyrosine glucosyl transferase. Tyrosine glucosyl transferase. Next, because the reaction is catalyzed by glycogen itself, that means what? Via autoglycosylation, glycogenin is also considered as an enzyme. Okay, and what happens to this? Glycogenin then catalyzes the transfer of up to 7 additional UDP glucose supplied residues, forming a primer. So this is the glycogenin. Under the influence of this enzyme, tyrosine glucosyl transferase, and even the glycogen itself, to the 194th tyrosine, 7 additional UDP glucose residues are supplied, and this acts as the primer. Okay, and the formation of primer starts the process of glycogen synthesis. So thus we require two things. The first thing is we require not just glucose 1-phosphate, rather than we require uridine diphosphate glucose for the synthesis of glycogen. And before the uridine diphosphate glucose donates glucose residues to the growing chain of glycogen, it requires a primer. A primer can be a glycogen fragment when glycogen sources are not depleted, or even it can be a protein called as glycogenin. And this glycogenin, on the 194th position, there is tyrosine residue, to which about 7 glucose residues are added, and this acts as a primer. Okay, so this is a glycogenin. On 194th position there is tyrosine. So tyrosyl 194 OH group. to which what happens? UDP is added. Then another one is added. How many added? 1, 2, 3, 4, 5, 6, 7 glucose residues are added by what we call the UDP glucose. So UDP glucose donates glucose residues releasing UDP. So each glucose residue is donated directly by UDP, not glucose 1-phosphate is not directly attached. It is converted into UDP glucose, and UDP glucose separates UDP and glucose, and this glucose is donated to the glycogen on the tyrosine residue. Then what happens? About 7 of these glucose residues are donated by UDPG, and thus a primer is said to be synthesized. Next, coming to the elongation or glycogen branching. Elongation and branching of glycogen is catalyzed by two enzymes. One is called as glycogen synthesis, the other is called as amylo 1,4,2,1,6 transglycosylase. Amylo 1,4,2,1,6 transglycosylase. So glycogen synthesis, it makes the alpha 1,4 linkages in glycogen and forms a structure called as alpha amylose. Whereas amylo 1,4,2,1,6 transglycosylase, as the name itself implies, it is responsible for the formation of 1,6 glycosidic bonds. Thus, it is responsible for branching. So, at branching, we have when we are discussing about structure of glycogen, at branch points there is 1,6 glycosidic bond, and the other points there is 1,4 glycosidic bonds. 1,4 glycosidic bonds are added by glycogen synthase, whereas at branching points 1,6 glycosidic bond is added by this amylo 1,4,1,6 transglycosylase. Thus, it is responsible for branching of glycogen. So, coming to see let us first discuss elongation followed by branching afterwards. So, glycogen synthase can add glucosyl residues only if the polysaccharide chain already contains more than 4 residues. Thus, we already have a primer with 7 residues. Even a glycogen fragment is there that should have more than 4. Let us see, it is also having 5. So, to this glycogen fragment, new glucose residues can be added. To this this primer containing 7 glucose residues derived from glycogen, also glucose residues can be added. So that what happens? Elongation of the chain takes place. Thus, new glucose units are added to the non-reducing terminal residues of primer. Next, the activated glucosyl unit of UDP glucose, the activated glucose unit, that means UDPG. So this is activated, we are saying this glucose is activated glucose. So this activated glucose unit of UDPG is transferred to the hydroxyl group. It is transferred to the hydroxyl group at C4 terminus of glycogen, to form an alpha 1,4 glycosidic linkage. So this is, we know that this is glucose 1-phosphate. This is glucose 1-phosphate. And here this is transferred to the hydroxyl group of C4 terminus of glycogen. So that what happens between these two, 1, 4, there is a linkage being formed. This is called as alpha 1,4 glycosidic linkage. So thus in elongation, UDP is displaced by the terminal hydroxyl group of the growing glycogen molecule. So glucose is converted to glucose 1-phosphate. Glucose 1-phosphate is converted into iridium diphosphate glucose. And now what happens? This glucose is donated by the iridium diphosphate glucose to the primer, during which this UDP is replaced by, this is the primer, this is its OH end, to which the glucose molecule is added. Thus, UDP is replaced by hydroxyl group. Okay. Thus, a new glycosidic bond is formed between the anomeric hydroxyl group of carbon 1, C1 of the activated glucose and carbon 4 of accepting glucosyl residue. Thus, a 1,4 glycosidic bond is said to be formed, and this is mediated by, catalyzed by an enzyme called as glycogen synthase. And this is the most important step or the most important enzyme in the synthesis of glycogen. and hence it is called as a regulatory enzyme. Okay, so this is UDP glucose, and UDP is removed, and to the existing primer, to the OHN, what happens? Another glucose is said to be supplied. So these glucose residues are added. Thus, what happens? A glycogen is elongated. And coming to the branching. So a linear form of glucose residues we have already uh, initiated the synthesis. Now at branching points, a branch has to be formed. So let us see how this branching occurs. So glycogen is a highly branched tree-like structure. Branching makes it more soluble. Why it is highly branched? Because it makes it more soluble and also increases the number of non-reducing ends to which new glucose residues can be added. Okay, so as more and more ends are non-reducing ends are present, new glucose residues can be produced and more amount of glucose can be stored by this branching pattern. Thus, the process of glycogen synthesis is accelerated. Okay, glycogen synthase can add glucose residues only if the polysaccharide chain already contains more than 4 residues. That is what we have discussed. Glycogen fragment should contain more than 4, and the glycogenin protein also contains 7 glucose residue, a fragment with 7 glucose residues called as a primer. So glycogen synthesis can add glucose residues only to the primer. Then how the branching occurs? A branch is created by the breaking of an alpha 1,4 link and the formation of an alpha 1,6 link. So this is between these two there is an alpha 1,4 link, and this is broken and an alpha 1,6 link is formed between two glucose residues. Thus the glucose is said to be branching. The branching of the glycogen takes place. Branches are made by the action of the branching enzyme. Glycogen synthesis cannot do this. It can form only 1,4 glycosidic bonds, and whereas 1,6 glycosidic bonds at the point of branching is initiated by or catalyzed by the enzyme amylo alpha 1,4 alpha 1,6 transglucosylase. This enzyme, what does this enzyme do? This enzyme removes a set of 6 to 8 glycosyl residues from the non-reducing end of the glycogen chain. Thus, the alpha 1,4 glycosyl bond, glycosidic bond is said to be broken. So, breaking an alpha 1,4 glycosidic bond to another residue on the chain and attaches it to a non-terminal glycosyl residue by an alpha 1,6 like, thus functioning as a 4,6 transferase. So here it removes about 4 to a set of 6 to 8. A set of 6 to 8 glucose residues are removed, okay, from the non-reducing end of the glycogen chain, and thus alpha 1,4 glycosidic bonds are said to be broken down, and it attaches a glucose with for the formation of 1,6 glycosidic bond at the branching point. And thus the resulting new non-reducing end as well as the old non-reducing end from which 6 to 8 residues were removed can now be further elongated by glycogen synthase. So what actually happening? This is the glycogen, and let us say this is, and here about 4 to 6 are said to be removed between this place, okay, and from here what happens? A new chain is said to be initiated, and from here the old reducing end also new chain is said to be elongated. Thus, whenever this has to take place, and whenever the branching has to take place, it has to remove and then add the remaining. Likewise, a number of branches are said to be being formed. Okay, so this is the glycogenin. To the glycogenin, already primary is added, to which new glucose residues are added and elongation takes place. Whenever the branching is required, the alpha 1,4,1,6 transglucosyl, what is that? Transglucosylase is responsible for the formation of 1,6-glycosidic bond here, and from there what happens? Elongation continues. Thus a branch is formed here. So new branch can be formed like this on every branch. Thus a mature glycogen called as beta particle is said to be synthesized. So how about the synthesis of additional branches takes place? After the elongation of these two ends have been accomplished, after the elongation of these two ends have been accomplished, the terminal 6 to 8 glucosyl residues can be removed. The terminal 6 to 8 glucosyl residues can be removed and to make additional branches. So additional branches can be made by removing the same process, by removing the 6 to 8 terminal residues. Thus it results in the formation of what we call a mature glycogen, which is called as beta particle. Okay, so this is how the entire process of glycogen takes place. First of all, glucose enters the liver cell under the influence of insulin. It is converted into glucose 6 phosphate, and the same enzyme phosphoglucoamidase is responsible for the conversion of glucose 6 phosphate to glucose 1,6 bisphosphate, and then finally to glucose 1 phosphate. And this glucose 1 phosphate cannot take part directly in the synthesis of glycogen. Rather, it is converted into uridine diphosphate glucose with the help of UTP. Thus, uridine diphosphate, this blue color is nothing but glucose residue. Uridine diphosphate glucose is said to be produced. Okay, and this is said to be, this uridine diphosphate glucose is responsible for donating these glucose residues for the during the elongation and branching of the glucose residue. What actually happens? Glycogen synthesis requires a primer. Here, the glucose cannot be directly added to glucose. A polymer cannot be formed directly by the formation of glycosidic bonds. Rather, it requires a primer. Initial sequence of glucose residues is required. And where there are glycogen sources, here a small glycogen fragment can be used for this purpose. It should contain more than 4 residues of glucose. In the absence of glycogen sources, what happens? The body synthesizes a protein called as glycogenin, and this glycogenin protein adds 7 glucose residues. It adds 7 glucose residues, and this acts as the primer. So either a glycogen fragment or a glycogenin protein containing 7 glucose residues acts as a primer. And here these 7 glucose residues are added to the 194th tyrosyl OH residue. Okay, from there 7 are said to be added. Then what happens to this primer? To this is the glycogen fragment or primer. To this, the UDP glucose starts donating the glucose molecules. Thus glucose molecules are being added. So towards the end, what happens? 5 to 6 are said to be, 6 to 8 are said to be removed, and now a branch starts here. A branch starts here, and this branch is formed by the transglucosylase amylase enzyme, and this forms a 1,6-glycosidic bond. Whereas the glycogen synthesis forms during elongation 1,4-glycosidic bonds, and the second enzyme forms 1,6-glycosidic bonds. And if this branch is said to be elongated, and from the ends again 6 to 8 residues are removed, and from this branching point another branch can be added. So wherever a branch is required, there 6 to 8 residues are removed, and a new branch is initiated by the transglucosylase enzyme. Thus, a highly branched mature glycogen molecule called as the beta particle is said to be synthesized. So this is how, under the influence of insulin, glucose is converted to glycogen in liver, where it is stored mainly. And the same process takes place in the muscle also, and the glycogen is stored in muscle because muscles perform a lot of work rather than the liver. If muscles rely on liver for the breakdown of glycogen and supply of glucose, there is a brief period of lagging. And in order to avoid this, God made muscles in such a way that they can have their own source of energy in the form of glycogen. So this is all about glycogen synthesis. In next class, we will discuss about glycogenolysis. Okay. If you have any doubt, please ask me. Otherwise, we'll close. Thank you.